Today I presented some data on human haemopoiesis because that's what our lab works on, and I presented some data where we compared the composition of the human haematopoietic and stem cell pool in different organs, and in particular we looked at the bone marrow and the peripheral blood, and really the take-home message is that the composition of the circulating progenitor and stem cells is very different from that you have in the bone marrow, and that normally this is really skewed towards making erythroid cells, so it's a lot of progenitors that are circulating in the blood that are making red blood cells, but interestingly they're not making them in the blood. They are making them in the bone marrow, but they circulate in large numbers in the blood. And the second thing I've been discussing is really how long-term HSCs, which are the cells that are responsible for the outcomes of transplantations in patients, once they are put in culture, they are really experiencing a very strong stress of the culture. They need to adapt to this culture and they lose their stem cell function. And while this has been known, what we've been able to show is that this loss of function occurs very early on, and unlike what people were thinking of before, it really is it's not related to their cell cycle progression. It's not due to the fact that they divide, but it's due to the fact that they really change their microenvironment. And I think this is effectively good news because it means that we should be able to expand stem cells ex vivo, and it also gives us new opportunities to target these very early events that occur molecularly to potentially improve many different applications which require cultures of stem cells.